Great. Well, it's a great honor to be invited to give a talk at the MVIF. I've been a fan from afar for quite a long time. These types of virtual events, I think, really knit together a super vibrant and very international community of microbiome researchers, and it's a critical thing to have, I think. So, without too much more ado, I'll just jump right in because Serena gave a really great introduction to my lab's work and how we're thinking about how we can engineer the microbiome to our benefit. So I'll talk today about kind of our work for the last few years in understanding the metabolic outputs of complex microbiota and how we might start to think, have the hubris maybe even to start to engineer those outputs. So I'm sure none of you need this introduction, but of course, we are mostly microbial. We've all heard this many, many times, I'm sure. We're about on par by the numbers, right? There's about half as many microbial cells in and on our bodies as human cells, coating our outer surface, although they're much smaller than our cells, 1,000 times smaller than eukaryotic cells. So it's only a few hundred grams of material. But the genetic capacity of this community vastly outstrips our genome. Right? We have 23,000 genes or so, and we have something like 4 to 6 million unique genes in our microbiome. And a fair amount of the functional capacity of our body system has been outsourced over evolutionary time to this complex ecology. And there are many things that make us all unique individuals. One of those is our genome. However, if you have an identical twin, you do actually share a genome with someone else in the world. But even identical twins have completely distinct microbiota, right? Two identical twins have gut microbiota that are as different almost as two strangers. Not quite, but close to it. And so it's this hugely important factor that's affecting our personalized responses to interventions, to diet. We're just starting to get a handle on it, right? And I would argue that, one of the major ways by which the microbiota influence human phenotypes and physiology is through the production of small molecules or metabolites. And we know that if you look in the bloodstream and do an untargeted metabolomics and look across many, many people, a fairly large fraction of circulating metabolites in the blood are significantly associated with cross-sectional variation in the ecological composition of the microbiota, right? We found about 44% of all blood metabolites showed some significant association with the microbiome. But how do we integrate all of this complexity, right? There are thousands of metabolites. There are hundreds of bacterial species in the human gut. There's this conversation going on between our tissues and this complex ecology, and we don't yet have the Rosetta Stone that allows us to decipher what is being said by the microbiota to our bodies, or vice versa. And so that is the task ahead of us. How do we measure this metabolic function? I'll quickly give you a little philosophy there. There are lots of ways you can look at it. You could look at the genes, sequence the metagenome. You could look at proteins, proteomics, what's actually expressed from that genome. You could look at the metabolites themselves that are the byproducts of the enzymes that are encoded in an organism's genome and gets kind of snapshot concentrations of those metabolites. But I would argue that we should be really interested in metabolic flux, which is what this cartoon or this little video is showing you, is the actual mass movement of material through a metabolic network. Right, a given metabolite at a snapshot concentration might actually be very, very low abundance. And it might look unimportant, but it might be produced at an incredibly high rate and it might be consumed at an incredibly high rate. It could be a completely central node in this network and you wouldn't know it by just measuring a snapshot. So having this sort of dynamic view of flux, I think, is really important. But it's really tough to measure that flux in a real world system. You can do stable isotope labeling or time series measurements of concentrations, but it's tricky. Luckily, though, there are computational methods that enable us to try to estimate this network of fluxes in these complex biochemical systems. What these methods require is a pretty detailed knowledge of metabolism, and luckily over the last century, we have accumulated quite a vast trove of knowledge of central metabolism for both, mammalian or eukaryotic organisms and prokaryotes like archaea and bacteria. So I will be talking today about methods that rely on something called flux balance analysis. And this is essentially a formalism for encoding a biochemical network into a system of equations, in particular kind of a system of linear algebra, where we have matrices and vectors. So you have a stoichiometric matrix S and a flux vector V, and I'll give you an idea what those look like. We make an assumption, the steady state assumption, which is that S times V equals 0. So the sort of influx of material and the outflux of material are balanced. Like when you open a window in your house and you open another window on the other side of your house doesn't fill up with air and explode, right? There's an equilibration of the pressure, air flows through, and we sort of assume the same thing is going on in these metabolic systems. We can bound and constrain these fluxes with knowledge like thermodynamics or the availability of metabolites in the medium. So this is often also called constraint-based analysis or modeling. To give you an idea of what this S matrix looks like, this is a very simple biochemical reaction network with reactions R1 through R6, and metabolites A, B, C, and D. We can encode this network into a matrix, which you see here on the right. So here the columns are reactions, the rows are metabolites, and if you look at reaction 2, for example, it consumes A and produces B, -1 and +1. And indeed, if we look at reaction 2 over here, it's consuming A, it's producing B, just to give you a feel for how the math is done there in encoding these networks. What we want to estimate with FBA is this flux vector V, which is the number that tells you the amount of mass flowing through a given reaction in this stoichiometric matrix. And to how you find solutions in these really big high-dimensional systems, it's tricky and it's complicated. This is just a pictorial example of, this polygon is showing you the allowable space given this steady state assumption, S times V equals 0. And if you do something like maximize or minimize, you can sometimes find unique solutions. It relies on some convexity to the space. So for example, if you maximize flux through V5, you can find a unique solution here. Maximize through V6, you can find a unique solution there. And usually these are much higher dimensional systems. And what we're often trying to solve for is the maximization of biomass production. We sort of assume that bacteria are trying to grow as much as they possibly can in these steady state models, which is a reasonable assumption given some constraints. Okay, so that's a little background on FBA. FBA traditionally was applied and created for a single organism, E. coli growing in a bioreactor. And what is only now in the last a decade or two begun to happen is to the extension of these types of models to more complex multi-species communities. And so that's what I'm going to talk about today. And we see these as kind of the Rosetta Stones that we are seeking for trying to decipher ecological composition and ecological function, right? How do we map who's there to what they're actually doing? And this picture here is just showing you an example of one of these large community scale metabolic models that we've constructed. This is one person's microbiome represented as a flux network. These regions of high density are individual organisms with a lot of internal fluxes going on inside the cell. You see some cross-feeding interactions across the boundaries of these cells, and then there are large swirls of kind of extracellular metabolisms that are occurring in the system. These are very, very big models, tens of thousands of reactions and metabolites and hundreds of thousands of connections. And so in the last few years, I had a really talented research scientist in my group, Christian Diener, who is now an assistant professor at the Medical University of Graz in Austria. And Christian is just a amazing metabolic modeler, super skilled guy and part of the Cobra Pi community. And he wanted to tackle this problem of building a tool that enables the construction of these MCMMs, we call them, microbial community scale metabolic models. And this was also done in collaboration with Osvaldo Resendez-Antonio, one of our collaborators. So you may have heard of it. It's called MyCom. It's a portmanteau of microbial and community, and it just allows you to build these models. as inputs for these things, what you need is essentially a constraint on who's in a person's gut microbiome. So you kind of sequence their microbiome, you see what taxa are there, you map those organisms to a database of curated metabolic models like Agora. And then another constraint in order to build these models is you need an input diet that you can feed to the microbiome. We use the virtual metabolic human because they have about a dozen pre-computed human diets that we can leverage. We also do some adjustment to those diets because we assume that a lot of the nutrients in a human diet are absorbed in the small intestine. So we use the human Recon 3D metabolic model to extract those metabolites that are absorbed by humans, and then we feed the rest to this colonic milieu where the microbes are growing. We use some tricks to do the optimization here. The name of the game for us here was balancing between speed and accuracy. If you want to be really, really accurate, these models with tens of thousands or hundreds of thousands of edges will take, days, if not years, to find a solution, right? So we want to be a little faster than that. So we pulled some tricks to get that to work. I won't get too deep into the details there. At the end of the day, we get estimates of the cross-feeding interactions between microbes, the net influx or uptake of metabolites from the medium, and the net production of metabolites from the community that are available to the host. So if you want to use that or read that paper, that's the citation. We recently wrote a little perspective article in M Systems that explains some of our modeling choices for what we're calling cooperative trade-off flux balance analysis, or CTFBA. This is, like I was telling you, it's a balance between accuracy and speed. So we can get very rapid runs of our models. We can run thousands of models, iterate them, change little pieces, and then rerun them. And it allows us to get fairly accurate output. The last thing I will say about it is that it approximates a dynamic trajectory. There are methods called dynamic FBA that are out there where you can simulate from the point of no growth to the kind of steady state and back. What CTFBA does is it guarantees that you're going to have the shortest path between the point of no growth to the sort of steady state solution, and it's going to estimate that point for that shortest path, essentially. Okay. So does it work? Is this something that we can actually use? And here's some information to kind of give you an intuition. So if you ran one of these large community scale models naively and you optimize for community biomass production, which is kind of a way to get a big speed up in your modeling, as opposed to giving every single organism their own optimum, what you find is that everybody kills themselves so that a few taxa can grow. This is not a terribly evolutionarily realistic outcome, but it is what happens when you naively run these models. With our sort of cooperative trade-off, where we have this L2 penalty and then a slight reduction in the maximum for the maximal community biomass level that you can achieve, we start to see that we can achieve a larger fraction of the community that can grow. And we usually have some cutoff where, above some threshold that makes sense for the system. So for example, in the human gut, between 70 and 80% of all cells are alive in a given metagenome. And so if we can get something like 70 to 80% of everything growing in the system, we're pretty happy. So we try to pick a minimal trade-off that gives us this kind of growth behavior we expect. It's a bit heuristic. We do have some first principles for how we do this, and that's embedded in the perspective I talked about a second ago. Another way we can validate the outputs of these models is to directly estimate growth rates from a sample, like a metagenome, using replication rates. So this is a peak to trough ratio thing. I don't know if you've heard of it, but it's a cool method where microbes have parallelization of their replication forks. And if you pop open a fast-growing cell, it's going to have a lot more DNA near its origin of replication than near the terminus. And if you map those reads back to the reference genome, you're going to get a slope. And the steepness of this slope is proportional to how fast that organism is replicating its genome. So we can get this estimate directly from a metagenome, and then we can compare those estimates to our model inferred growth rates. And what we find is that if you don't run our method at all, you get essentially zero correlation between these measured replication rates and estimated growth rates. But if you do our method, you actually get reasonable validation, not perfect by any means. Okay, so what, assuming we're doing a decent job here of simulating the system, what kinds of things can you do with it? This is just an example of one person's metagenome where we've knocked out every single organism one at a time in their system, and we've seen what that does to the growth rates of all the other taxa in the system. So for Bacteroides, for example, if you knock it out, a lot of other things grow more quickly. So it seems to have a suppressive effect on growth rates. Whereas Akkermansia is the opposite story. If you knock it out, a lot of other things grow less well. So it seems to stimulate the growth of many other community members. But this is really fun to kind of explore these complex communities and do in silico experiments. It would be really, really tough to do in reality. But the question that arises is this useful for anything? Could we actually leverage these types of models to make translationally relevant predictions? And so that is what I'll now focus on. So this is work that was recently published back in the summer, in July of this year. Work driven by a really talented grad student in the group, Nick Quinn Bowman, who's now Dr. Nick, and a huge team of folks because there was a lot of experiments and collaboration here. So Kat Ramos, Crystal Perez, Christian, who you've met, and then Alex Carr, another grad student who's now Dr. Alex, who's graduated this last few months. In collaboration with our collaborators at Fred Hutch, Joanna Lampe, and Lisa Levy, we applied our models to predicting short-chain fatty acid production. Many of you probably know short-chain fatty acids are these critical molecules that our microbiota produce that are available to us in our colons, and we're probably underexposed to them in the developed world because we have lower fiber diets than we used to eat. But our bodies expect to see these organic acids, and if we're depleted in them, health problems arise. But the problem is, if you feed 10 people a banana, the same amount of fiber, you get very different amounts of these SCFAs out the other side. And there was really no rational way that we knew of to predict this before we tackled it. So that's what I'll talk about. In particular, I'll focus mostly on butyrate, which is the most potent of these short-chain fatty acids. And if you're not familiar with butyrate, it has widespread effects throughout the body. It's largely produced in the periphery, in the colon. It's rapidly absorbed and degraded by colonic cells. It's the primary carbon and energy source for colonocytes. It's also a signaling molecule, so it can stimulate enteroendocrine cells to produce GLP-1, which you've probably heard of, this appetite-suppressing hormone. It also is an HDAC inhibitor, so it affects epigenetic decoration of host cells, so it can bias T cell differentiation. More butyrate means a larger number of T regulatory cells in the gut, which dampens inflammation and usually leads to kind of lower systemic levels of inflammation. And higher levels of it generally has been associated with better health. But how do we validate our model predictions? Like I said, it's really hard to measure these fluxes in vivo. You need like a little robot in someone's gut to be able to continuously measure the production of butyrate because it's being consumed and degraded almost instantaneously as it's made. So what we did in order to get these measurements of flux outside the body is we get poop. So this is our collaborators, Joanna Lampe and Lisa Levy at Fred Hutch. We get a donor, they call us, the poop drops. Within 20 minutes, we get the poop into our anaerobic chamber. So really fast. We turn that poop into soup, but the more professional way of saying it, I guess, is an ex vivo, where we kind of homogenize the stool. We put it into 96 well deep well plates, or here's a beaker, just as a cartoon. And then we can add interventions to these soups. So we can add a little bit of dietary fiber or just do a control intervention of PPS buffer, incubate for just a few hours, take a baseline targeted metabolomics sample and then an endpoint sample. And this allows us to measure a flux. We can actually empirically measure the production flux of propionate or butyrate or whatever molecule we want to measure. So we did this, and then we compared these measured fluxes to our model fluxes. We can do the same experiment in the model. We take the same person's microbiome that's been sequenced, we put it into the model, we add the same fiber, we predict the butyrate production, and we compare that to the measurement. So when we do that, this is essentially a summary of all the different experiments that were run for butyrate and propionate, we get decent prediction. Our R value is about 0.6, right? Not perfect, but decent. One thing I will mention with the model predictions here is that there was no statistical fitting done here, right? This is a purely mechanistic model. That's a purely mechanistic prediction. So we expect to underperform relative to a well-trained machine learning model. But if we want to compare this level of prediction to some other work that's been done, I found it kind of edifying to go back to this Zivi et al. Paper, which you probably are familiar with. This is from Eran Segal and Ron Elenov's group, where they had a large cohort of Israelis who measured their diet and their microbiomes and some clinical chemistries, and built a gradient-boosted decision tree model to predict post-meal glucose responses. And this is what they get at the end of that. So they have model predicted versus measured, and their R value is about 0.68, right? So it's not terribly far off from where we're at, although they have much more data than we were able to generate. These ex vivo experiments are quite expensive to run. Okay, so in vitro, our model is fairly well validated for some of these molecules. What about their putative effects in vivo? So we did have data from a large population, more than 3,000 people, with paired microbiomes and paired blood chemistries or clinical labs, the kinds of things you get measured at the doctor, like your LDL cholesterol and so on. And we could ask, of the 128 clinical chemistries that we had measured, how many of them were associated with butyrate or propionate production from our models? And this is generally what we found. So these are the set of labs that were significantly associated with one of these three columns. And it turns out that only the middle column showed any significant effects. So these stars are denoting statistical significance. And this is the model predicted butyrate flux. We saw no associations with propionate, interestingly. And we also had measurements of butyrate from the blood. So we had blood metabolomics, and there was actually no association at all between blood levels of butyrate and any of these clinical chemistries, which is kind of fitting our intuition. We think that most butyrate is being kind of destroyed or consumed at the periphery, and very little of it is making its way into the bloodstream. And what little is there is very kind of homeostatically controlled by the host. To give you a feel for what these different chemistries are, it matches our intuition. So these are all the kind of lipid health measures, so like LDL cholesterol or triglycerides. And generally, if you're predicted to make more butyrate, you tended to have lower levels of these analytes in the blood. Same for insulin resistance. So markers of insulin resistance, you tended to be more insulin sensitive if you produced more butyrate. And finally, inflammation. So CRP, this is a measure of systemic inflammation. Those people who produced more butyrate showed lower levels of blood CRP. So that's kind of good. I mean, it sort of matches our intuition for how we think butyrate should be behaving in vivo. And we think that our model predictions are actually more resonant with these phenotypes than measurements from the blood or even from stool. So, okay, imagine that this is working, right? What can we do with it? So we played a game where we could flip this population between two different diets. So European, which is kind of a low fiber American style diet. It's based on the Austrian diet, and a high fiber diet. And we noticed that, yes, we generally get higher butyrate production on this high fiber diet, but there's a subset of people that are non-responders that just don't increase. And there are even a few regressors, so people who show lower butyrate production when they're given the high fiber diet. So we decided to implement a few very simple co-interventions on the backgrounds of these diets, two different prebiotics, inulin or pectin, and then a probiotic, Faecalibacterium presnitzii, which is a prevalent butyrate producing microbe in the human gut. So when we do this, this is the sort of summary of what we find. It's a heat map showing butyrate production as the heat. Every column is an individual person. The regressors are here, the non-responders are here. The black boxes are outlining the intervention that was optimal for that individual person. And if you just glance across the plot, there's no single intervention that was optimal for everyone, right? Different people needed different things. For some folks, just adding a simple prebiotic fiber like inulin or pectin was able to turn up their butyrate production. But for others, maybe they didn't have the genetic capacity to produce much butyrate. So it doesn't matter how much feedstock, how much fiber you fed down their system, they're just not going to make that much. So they required a probiotic intervention in order to respond. And so we're kind of developing out this type of modeling to see if we can prove this type of thing in a prospective clinical trial, which we'll talk about at the end. And given my time, I'm just going to whiz through to the next story, which is, we're also trying to think about adding personalized dietary constraints to these models. Currently, we're making a very dumb assumption that everybody's eating the standard American diet or the standard Mediterranean diet. It's actually hard to get individualized dietary constraints for this modeling. So we wanted to think of an automated way that we could do that. So Christian worked on a method that is currently in review. Hopefully it'll be out soon, where we are trying to infer individual dietary intake from residual DNA from plants, animals, and fungi that we eat that is showing up in stool. So we call this method MEDI, or metagenomic estimation of dietary intake. One of the first challenges is we had to build a large database of all sequenced genomes of plants, animals, and fungi that are present in the human diet. So this is just a view of that tree. Another challenge is we had to map the taxonomic information of those organisms to something about the metabolic composition of their biomass, right? Like, can we also estimate nutritional intake in addition to the types of foods that are being eaten? And so we use FoodDB for this. This is actually a database that does this. And we were able to find genomes for 91% of all the foods represented in this FoodDB database. So how do we test this? There are various ways we played around with testing the model. One was in silico, so we can produce sort of in vitro metagenomes. We took the sort of HMP2 data set as a kind of reference for gut microbiomes, kind of randomly subsampled individuals from that, created artificial metagenomes where we either had no food reads included in those metagenomes. So this is the negative control. It's just human and bacterial and viral and archaeal DNA. And then we had positive controls where we added 10 different food items at random from a large set, distributed as sort of exponentially in their abundances. So if some food's really abundant, some food's really rare, and then looked whether we could detect them. First off, I should say, I forgot to mention that this is like a really huge database of genomes, and searching it is very costly in time and effort computationally. So we had to kind of use fast methodology. So we use Kraken-2 to essentially build a hash of this large database and use K-mer-based methods to do this search. But those are really prone to false positives. So we had to take care of that, right? You might have seen the cancer microbiome paper from a while back. They ran into some of these issues, and we learned from that paper what not to do. So we had to put decoy genomes for all the other organisms in the system, like human genome and all the bacterial, archaeal, and fungal genomes, viral genomes from RefSeq, all the plasmids. We just pulled everything we could as a decoy to kind of pull away those annotations. We also had some entropy filters. So for example, some reads will have multiple different annotations from across the phylogenetic tree. Those are garbage, so we throw those away. But at the end of the day, we get a pretty good false positive and false negative rate. False positives tend to be organisms that are phylogenetically related to a taxon that's truly in the system. So say species within the same genus of plant, for example, like Brassica, which is kind of okay because those organisms actually tend to have similar nutrient content. We can also validate our methods experimentally. So we teamed up with a few researchers who had controlled feeding studies. So this is Hannah Holscher. Hannah had a study where they added avocado to one arm of a dietary intervention. So one group of people got avocado, one group didn't. If we run MEDI on these metagenomes and ask what's different between these groups, only one thing, avocado. That was pretty good. We can also look at nutrient predictions. So we can predict the amount of energy or potassium or total carbohydrate, total protein. And generally we get pretty good agreement between the dietary records or questionnaires and the predictions from Medi if you normalize to a 100-gram portion. But there are some places where it breaks. So dietary fiber, it breaks, total fat, it breaks. And we think this has something to do with processed food intake, because processed foods tend to be depleted in fibers, whereas the whole food, Medi is assuming that you're eating the whole food, essentially. So we need to do some correction to fix that. This is just an example of what a profile looks like from a bunch of different people. It's a heat map of all the different food items detected. In this population, the most common detected food item is wheat. This is the Latin name for wheat. The most common meat was pork. This group here are the apple eaters, right? The Malus genus. So you get really rich information. You can use this to kind of assess differences between health states in cohorts that don't have available diet data. So here in the metacarditis cohort, there were healthy people and people with metabolic syndrome. We didn't have dietary questionnaire data, but if we asked, what's differential in Medi between these two groups, we found that people who had metabolic syndrome, they ate more meat, they ate more pork and chicken, and those who were metabolically healthy, they actually tended to eat more veggies and fruit. So I'm moving fast because I know I'm behind on time. We're working on figuring out the biases to better kind of create diets that can be fed into our metabolic model. We're thinking about ways we can use reference populations, eating known diets, have some unknown sample, calculate some sort of beta diversity distance, and use that to weight some kind of dietary vector that we can then use to constrain the model. So Nick is working on this. That's all TBD. And then in my final few minute or two before I end off, we're thinking about what's next for this type of modeling. We've done a fair amount of in vivo and in vitro validation, and a lot of these interventions that we want to do are pretty safe, right? We just want to feed people like bananas or probiotics. So I feel like we're ready to move to the clinical trial stage to try to test some of these precision interventions and see if they perform better than placebo or if they perform better than standard of care, or what have you. So we are working on a platform that we're calling My Digital Gut. And today it was just announced we got this AWS Imagine grant to help build this prototype. And to give you a feel for it, we have some mock-ups, but here's like John and Jane with their logins. This is a sort of a decision support system that we're trying to build for clinicians, researchers, and maybe even for individual people eventually, if we can validate that this is working. The idea would be, pick some sort of high value metabolite like butyrate that you want to optimize. You can simulate people on various diets, prebiotics, probiotics. You can compare their levels to some population distribution, and then make choices for yourself or make choices for an individual in a clinical trial. The direction we want to go initially is trials, right? We want to show efficacy for specific indications like reducing prediabetes or optimizing cardiometabolic health, things like that. But eventually we want to make this into some kind of decision support system, right? If it's validated and it's working, we want to provide clinicians with a simple way to deploy precision nutritional interventions that leverage the microbiome. And then maybe, further down the road, years later, this could potentially be some sort of direct to consumer thing that's available. So I'm sorry for whizzing by so quickly. I'll thank all the people involved in this work, all the micronauts in my lab who do the work. Thanks to the funders, the collaborators, and thanks so much to all of you for listening.